The History of Antibiotics. Back in 150 AD, way before the advent of the antibiotic age, Claudius Galen was a very well-known physician and surgeon. He was very influential when it came to medical ideology during this time. His major medical theory, known as the four humors theory, came from the idea that sickness came from an imbalance in the four humors, which were blood, phlegm, black bile, and yellow bile. His treatments revolved around rebalancing the four humors. Common treatments included bloodletting and the use of enemas. At this point in time, all medical treatments were either palliative, meaning just making the patient comfortable, or they were very aggressive and sometimes deadly in and of themselves. Another important theory at this time was the miasma theory. The miasma theory involved the idea that rotting and decomposing material made the air around it bad and could cause sickness when breathed in. It is also important to note that the idea of quarantine was used very long ago, even before the idea of the four humors theory came along. People that had leprosy were often isolated from everyone else in fear of the sickness spreading to others. Fast forward to 1843. In London, people seemed to be getting very ill from what would later be identified as cholera. John Snow, now known as the father of epidemiology, noticed this and carried out the first ever double-blind epidemiological study. He knocked on people's doors randomly and asked whether anyone in the home was sick, as well as what water pump they used to get water. Snow eventually figured out that those who were getting sick were getting water from the same pump, and that this water was being contaminated with feces that was spreading the disease. The miasma theory, the use of quarantine, and John Snow's discovery foreshadowed the germ theory of disease, which was published in 1890 by Robert Koch. By carrying out a series of experiments, Koch was able to come to four conclusions. 1. The microbe that causes disease is found only in sick and not healthy organisms. 2. The microbe must be able to be isolated and purified. 3. The microbe must make a healthy organism sick. And 4. The microbe must be able to be re-isolated from the now sick organism. Now, the goal of medical interventions had changed. The new objective was to get rid of the microbes, or germs. During the Industrial Revolution, many dyes were being used. Paul Ehrlich realized that some dyes would only stain specific cells and not stain others. He hypothesized that if dyes could target specific cells, maybe a dye derivative could kill specific cells. This led him to look for the first chemical therapy. In 1910, Ehrlich introduced the drug arsphenamine, which was sold under the name of Salvarsan. He found that arsphenamine treated syphilis, which was a big deal considering that syphilis was very painful and people of all socioeconomic classes would contract it. It quickly became the most widely prescribed drug in the world. However, it is important to note that it was not very easy to administer to patients as it was not very water-soluble. It could also result in very severe side effects, one of which was limb amputation. In 1912, Paul Ehrlich developed another drug called neosalvarsan, which was very similar to salvarsan, but it was more water-soluble. Neosalvarsan quickly overtook salvarsan as being the most effective treatment for syphilis. However, it still had some serious side effects. In 1928, Alexander Fleming and his assistant Stuart Craddock were conducting experiments on the bacterium Staphylococcus aureus. As they were conducting their experiments, some of their petri dishes got contaminated with penicillium mold. There appeared to be a lack of bacterial growth surrounding the mold. Fleming and Craddock ultimately had two key discoveries. The first was that penicillium secretions kill some bacteria. The second was that penicillium secretions are harmless to animals. However, they struggled conducting further experiments on the penicillium mold because they were having a lot of trouble producing a large amount of it. In 1937, the Dunn School team at Oxford University began to investigate penicillin. This team consisted of Howard Florey, the group leader, Ernst Chain, the chemist, and Norman Heatley, the mechanical genius. When World War II broke out in 1939, the Dunn School team worried about their work being destroyed if there was an airstrike and they had to evacuate. So they spread the penicillium mold on their lab coats. This way they could easily restart the project if their lab got destroyed. In 1940, the Dunn School team published a study entitled Penicillin as a Chemotherapeutic Agent in The Lancet, an English medical journal. They debated as to whether or not they should share their methods in the study, but ultimately decided to share for the greater good. Their study consisted of giving people who were very close to death penicillin. Initially, it had bad side effects. However, if they purified the penicillin with chromatography, it seemed to help. This study involved a lot of work. Ernst Chain began working with Edward Abraham to try and make more penicillin. One of their methods involved freeze drying. Norman Heatley had the idea to collect and sterilize bedpans from the local hospital as a way to make more concentrated penicillin. In 1941, Albert Alexander was the first patient treated with penicillin. He was a police officer and managed to scratch his nose on a rose thorn. This scratch got infected and resulted in his hospitalization. Although the penicillin worked for a little bit, he ended up dying because there was not enough penicillin available to completely treat him. In July of 1941, Howard Florey and Norman Heatley came to the United States to meet with drug companies in hopes of getting help to figure out how to produce penicillin in larger quantities. However, none of the companies were interested in helping them. Finally, they met Charles Thom, who had connections with the Northern Lab in Peoria, Illinois. At the Northern Lab, the Dunn School team had three main goals. The first was to find a strain of the mold that produced more penicillium. The second was to optimize growth and production conditions, and the third was to greatly scale up production. The goal of finding a hyper-producing strain was met by Mary Hunt, also known as Moldy Mary. She happened to find it on a cantaloupe at a fruit market in Peoria, Illinois. Andrew Moyer successfully found a media that allows for the production of 1,000 times more penicillin. This media was made of corn-steep liquor and sugar. Robert Cogill established the method of deep tank fermentation, which allowed a large quantity of penicillin to be produced since sterile air was emitted into the large tanks, and the entire area of the tank could grow penicillin. In February of 1942, Ann Miller was the first patient that was successfully treated with penicillin. She lived on to be 90 years old. In November of 1942, penicillin got a lot of publicity. A nightclub in Boston called Coconut Grove was accidentally set on fire. Many people were injured in this fire. Most of the patients were given both sulfa drugs and penicillin. However, it is very likely that the amount of penicillin given to each patient wasn't enough to make a difference. The Boston Globe published a paper calling the drug priceless penicillin. In September of 1943, a war production board was established. This board basically tried to get companies to produce penicillin. Some of the incentives to produce penicillin included tax breaks, the ownership of any patents and further developments, and the government guaranteeing profit by agreeing to buy the penicillin. In 1945, Dorothy Crowfoot Hodgkin successfully figured out the structure of penicillin through the use of X-ray crystallography. At this point, you may be wondering why Alexander Fleming seems to have gotten all the credit for penicillin. Although his discoveries were monumental, without others, especially the Dunn School team, it would have been rather useless. The reason that the Dunn School team does not get as much credit is due to a publication in the Times of London in August of 1942. It told of the work being done on penicillin, but did not name any of the scientists working on it. Almroth Wright, who was Alexander Fleming's mentor, submitted that Fleming was the one doing all of this work. By 1944, penicillin was being mass-produced. However, in 1943, another antibiotic called streptomycin was discovered. One of penicillin's weaknesses was that it didn't work against all bacteria, specifically Mycobacterium tuberculosis, which is especially dangerous. Streptomycin was discovered by Albert Schatz and Selman Waxman. Schatz was a graduate student working under Waxman at Rutgers University. The lab was sponsored by Merck. Waxman developed a technique of systematic screening in order to find antibiotics that would work against tuberculosis. In late 1944, Patricia Thomas was the first person to be treated and cured of tuberculosis. Waxman got incredibly rich from the royalties and ended up receiving the Nobel Prize in Physiology, but he did not mention Schatz in his acknowledgments. This resulted in Schatz suing Waxman. Following penicillin and streptomycin, many other antibiotics were discovered. In 1945, aureomycin was discovered, which was the first broad-spectrum antibiotic. Another compound called terramycin, which was very similar to aureomycin, was discovered soon after. Robert Burns Woodward was able to find that they were actually mildly different compounds, which was significant because a different patent was available for terramycin. In 1945, another broad-spectrum antibiotic called chloramycetin or chloramphenicol was discovered. Its simple structure made it very easy and cheap to be synthesized in a lab. In 1949, an antibiotic called erythromycin was discovered. Due to the many antibiotic discoveries during this time, the 1940s are known as the golden age of antibiotics. The monumental discoveries of antibiotics in the mid-1900s carry great significance today, as antibiotics are used very often. However, it is important to note that the issue of bacteria developing resistance to the antibiotics is a problem that is already happening and will continue to happen until a solution is found. This can create massive issues in the medical field and warrants further research and hopefully further discoveries.